Yeah. So hello, today we are here for the verification seminar of eBMS. This is under the project of Embrace that we want to share with you how to use this tool and how to use it on eBMS and some practical examples on how they are using it on some countries in Portugal and in Denmark specifically. Well, as David say, we are going to present, well, he's going to present the presentation he did in Wageningen in the eBMS coordinator meeting, but well, it's good to remind some things and to get it better, subtle. And I will do later explain how is exactly the verification system and the different buttons and so on. So yeah, go ahead, David.
Great. Thanks, Christina. Yeah, so this will be familiar to some people who were there in Wageningen, but I think there's some people who weren't there. So yeah, I was going to give an introduction to the validation and verification approach that we have available in the eBMS system through the butterflymonitoring.net website, which links to the Butterfly Count app. So I think it was just quickly sort of doing some terminology in terms of the difference between validation and verification. So I think the validation is just ensuring that we have good standardized data that's complete and accurate. And that often means things such as dates are correct. We don't have 30th of February, those sorts of mistakes that can creep in. We have good geographic information, standardized information on the count, time, etc., and associated information such as images. Verification is really around then deciding whether the record that's been submitted is correct in terms of particularly the identification of species being submitted. So this is often using evidence such as an image, but also making a judgment based on where a species has been recorded geographically, whether it's in the right part of the range and at the right time of year, etc. And obviously these two things are quite closely linked. To put a confirmation on a species record, you need to know information about when and where it was recorded, etc. And often verification, there's an element of also judging the expertise of the person undertaking the survey. So there are ways that we can also do that. So that's a general approach to the topic. And what we do in the eBMS system is I sort of describe it as the wisdom in the crowd. So what that means is that we work with particular experts in the butterfly and moth community in Europe who are assigned a role to assess the data coming through the system. So that's you sort of using the wisdom in the crowd. The alternative approach, which also can be highly successful, is wisdom of the crowd. And that's the approach that iNaturalist uses, where anybody with an account on the system can have a view on records. And then they use an approach where when there's a certain level of agreement, they sort of assume it's correct and reaches what they call research grade in terms of its quality. But the wisdom in the crowd, the approach that we take in terms of working with nominated experts, that's broadly the approach taken through observation.org, and many national portals, I think, around Europe use that approach. And in the UK, we have a system called iRecord that similarly uses that approach of working with nominated experts. And I just say a bit about iRecord because I think it shows that this wisdom in the crowd, although it's reliant on a few key people, it can be highly effective when you have the right tools and you have the right experts who are motivated to review lots of data for supporting their scheme or their use of the data. So iRecord is the national portal in the UK. As well as capturing opportunistic data, it's used for the UK butterfly monitoring scheme, and it's built on a set of tools called Indicia, which is an open source software for supporting biodiversity monitoring programs. And we have sort of largest data flows coming through that system, around 4 million observations per year, and about 2 million photos. So it's supporting a large endeavor and supporting a lot of users. Submitting data, around 24,000. And we work with around 700 experts who are the wisdom in the crowd, who in the UK context, in iRecord, support the review of data from all sorts of taxonomic groups, plants, insects, vertebrates, etc. And on the Lepidoptera side, we get it's the main platform used in the UK for butterflies and moths, including the UK BMS, through the support of Butterfly Conservation, who work with the recorders and the experts on the ground. So again, this just summarizes some of the figures in terms of the data coming through this system in the UK, around 30 million observations, around 13 million of those are butterflies. Moths is fewer because there's some legacy systems that people are starting to migrate now onto online systems. So I think this number will increase quite rapidly now. Some of these desktop UK systems are now sort of reaching end of life. But I guess the key figures on this plot are really around the number of verifiers supporting the review of data, around 130 experts looking at the butterfly data, almost 140 looking at the moth data. And it's sort of done on a regional basis in the UK. So this task can be made more achievable by bringing in teams of people to help review data, particularly on a regional basis when people know very well what butterflies and moths are likely to be occurring in a particular region. And it means that we get over 90, almost 100% of the UK BMS data is reviewed this way. And most of the opportunistic data is reviewed this way, 94, 97%. And again, a lot of the moth data is reviewed around 80%. And this is an ongoing process. So I guess we've got a lot of experience of this in a UK context for Lepidoptera, but also other groups. And the system that we've then adapted for the eBMS is using these same tools. So the advantage of the underlying software is it could be relatively easily repurposed for different systems. So you may be familiar, and this is where we'll spend a lot of time today, is going through the tools themselves. So it's accessible via the butterflymonitoring.net website. It's only accessible to those experts in the crowd, the wisdom of the crowd, that have been set up to do this role. So only those who've been assigned the role of verifier can see the page. But broadly, this is what it looks like. So assuming you've liaised with Cristina and I and been set up to have access, you'll have a page on the main menu called verification. And this is broadly the page that you'll see. And there's a lot going on this page. So inevitably, it's quite a complex task when you start developing this in more detail. And as people get to work with the system, there's requests for new ways of filtering data, or options for reviewing data, or corresponding with others who may have a view on the data. But this illustrates the main features of this page. As I say, we'll go through this in more detail and demonstrate it being used. But the main principles are, at the top of the page, is some ways of filtering the data. And that can be by default, the data grid that you see below, in terms of below the filters, the main body of the page, are records that are awaiting review. So once you've made a decision on a record, it disappears from this view by default. But some of these filters at the top allow you to look at data you've already accepted. And there's sort of some high-level, top-level filters at the top. And then below that is a tool for setting up more detailed filters. So that may be, you might want to filter on a particular species, or a particular geographic area, or a particular time period, etc. And you may want to focus on which I'll come on to in a minute. You may want to focus on records that have failed some automatic checks. So you may want to look at those which the automatic check suggests might be incorrect. So there's a whole range of options around filtering the set of records available for you to review. And then what's shown on the right-hand part of this screen is an individual record view. So once you select a record, in this case, the one highlighted in blue is the one that's been selected, you'll see a map of where it occurs, and the other dots on the map will be where this species has been recorded elsewhere in the region. And then you have a set of options in terms of your view on the data, what we call the verification controls. So that's broadly whether you accept or reject a record. There's a sort of category in the middle, which is you're not quite sure, which is called plausible. And there's some granularity in these options in that you can be confident and accept a record even though you haven't got a lot of evidence behind it. You may not have a photo, or you may not know the person submitting the record, but you generally think it's correct, which is the one tick option. And the two tick option is generally where you've got a lot of confidence. You've seen a photo, or you know the individual very well and their expertise, etc. But I think but broadly, in terms of us going on to use the data for other uses, it's really a accept or reject situation. And we can also see on the right-hand side, you can see the full details of a record in terms of all the information you would expect in terms of location, dates, etc. So that's the main parts of the page. As I say, we'll go through that in a bit more detail. I just wanted to highlight there's a range of tools which allow you to sign off and select a lot of data, a lot of records at once. And one of the main ways of doing this, which was introduced particularly for the 15-minute count data, which can be quite heavy data, because if you use that method in the app, it basically creates an occurrence record for every individual butterfly you've seen. So if you see 100 Magnolia in the field, it will create 100 records of Magnolia in the data set. So that can be quite tedious, ticking all those off. So there's a bulk verification option, which broadly says if you accept one species observation from this sample, a sample in this case is the whole 15-minute count, it signs off all the observations of that species from the sample. So in the case of 100 Magnolia, you accept one, and then it would accept all 100 of them as being correct, which is realistic in the case of a 15-minute counts, because you're surveying a relatively small area, so you don't need to look at every single precise observation. Yeah, this just shows it a bit more detail in terms of what I just said. So this is an example, a dummy example by my colleague. In this case, some swallowtail data, where you'd click on the bulk sample button, then you'd select an occurrence and then accept it. And whenever you make a verification decision, you can always add a comment as to why you've made that decision if you want to, but that's not required. So the other thing I just wanted to mention, which I mentioned briefly when I was talking about how you filter the data, is that we do have some rules in the background which are run whenever data comes into the system. They're actually run every 15 minutes because we don't run them, which is for performance reasons, we only run them every 15 minutes. But what this will do is we'll run a series of checks against every record that comes into the system, provided we've got these rules in place. And we do have these rules in place for the eBMS, for European butterflies. We don't have them for moths, but that might be an interesting discussion, how much we try and build that up. But the rule checks fall into three main types. The first of which, shown by icon from, it's supposed to be a microscope, but it's basically around the identification difficulty of the species. Actually, we haven't got this in place for the European data, so I'll skip over that one. But there is the option to have a rule based on the identification difficulty of the species. So you may decide in Northern Europe that every record of a brimstone, you think that can be broadly identified by anybody. So you might automatically accept all records of the brimstone, for example. So there's some rules that we can apply based on the identification of the species. Then the second main rule set shown by this globe symbol icon here is record checks that look at the known range of a species and would flag up anything that's been recorded outside of that known range. So that suggests that maybe this is a mistake, or it could be an expanding range situation, or it could be something occurring in a new location. But it's quite useful to flag these up as they come into the system, because you might want to check them in more detail. And then the other main record check shown by this calendar icon is assessing records against the broad flight period of species within a geographic region. So if things are being recorded early in the season or late in the season, they get flagged up. And all these checks at the moment, they're just used as flags on the records. So the results of these checks will be either a green tick, i.e. it's passed all the checks, or there'll be a red thumbs down, which suggests one of the rule checks has failed, and it will indicate which rule set has failed, either the geographic check or the time of year check. So you'll see those as you look at data coming through on the system. So it's really a way of enabling you to focus in on records that perhaps need most attention, because there may be errors and the records may be incorrect. So just to illustrate that in a bit more detail, I'm picking on the Danish BMS scheme records here, all records of Polymatus icarus. I think they probably all come from the same couple of locations shown on the map here. And using the rules we had available, they all failed the geographic rule checks. Actually, in this case, Andreas has reviewed them and decided actually they're correct. So the rules are not, we want to apply these rules automatically in this case. But it just illustrates what you will see if you look at the system and what the data shows in terms of records that flag up the geographic rule checks. And one of the slight issues with these is either they're clearly wrong. This illustrates another example where there were two records flagged up as being outside the known range. The one in Turkey, I can't remember the species. I think it's Aglis urtica, but I can't remember exactly which species it is. But the rules we're using are based on these range maps that Chris Van Swy created from GBIF and other data sources. I think may have been used in the red list, for example. But anyway, we've got maps of all species ranges, so we flag up anything that occurs outside them. So the one in Turkey is clearly outside the range. But actually, there is a slight issue here that the geographic boundaries aren't completely accurate in terms of how they're tied to the coastline. So in this case, in the Netherlands, there was an observation flagged as being out of range, but really it's only because of the quality of the underlying map data. So this is why we're using these really as flags rather than completely relying on some of these checks. And then the time of year checks, as I said, will flag up things which are either early or late in the season that we would expect. And this has been derived again by Chris, who's again used the data on GBIF and has applied an algorithm where for each species and each geographic region, biogeographic region that it occurs in, we have a date range. So the top example here, Agle canusa from the Alpine geographic region, we would expect it to occur mid to end of July. It's quite a narrow window. So anything that occurs outside that window will get a flag that it may be a mistake. And you'll see that the periods change for different geographic regions and species. Clearly, this is quite difficult to do in an automated way, but hopefully we've achieved a balance of not firing off too many checks and to really flag up things which are likely to be incorrect. So just a few questions to finish on from my perspective. I mentioned that there is the option of an ID rule, difficulty rule. So we could classify the butterflies by their ID difficulty. And we've done this in the UK actually, and I won't go into this in a lot of detail, but for the butterflies in the UK, we feel that most of them can be identified by a photo. But for the moths, it's much more challenging, and there's a set of species that need to be, only accepted from experienced recorders or need an expert to confirm. But we could do this for the UK, European butterflies on a regional basis. We've talked a bit about photos. The butterfly transect method and the 15-minute count don't really lend themselves to taking photos of butterflies because it's quite challenging when you're trying to count them. But particularly for the moths, we do have a lot of reliance on photos. And we do have a page, a verification page, where it's much more around reviewing photos. So the photos is the main thing that you see. So this is an example of the page, and you can tick them off more quickly, I think. So we'll introduce that. And then the final thing around photos is we do have access to the image classifier used on Ops Identify, built by Naturalis. That's already within the Butterfly Count app, and we will do some work to integrate the results of the image classification onto the online system so that it can support your view on the records. So typically what we see is the series of suggestions that the classifier thinks, and you can choose which one you agree with. Again, we're not using this in any automated way, but it's another way of supporting your work. Then the final thing I wanted to say is, I also mentioned this in Wageningen, that we do have a mechanism to, once you've reviewed data sets and you're confident in that they're generally correct or very confident if you've reviewed all of them, we have a mechanism to publish data sets automatically to GBIF. The example that is already in place is from the Denmark scheme again, where they publish data sets for the transects and the time counts on an annual basis, having reviewed the data. So I think that's it from me, Cristina.
Yeah, thank you so much, David. I think it was pretty nice introduction and explanation of all the icons. I don't know if there are questions up to here or... Yeah, Andreas.
Hi, David. A question, number one, moth verification. I think it doesn't work for us, yes, at the moment. I know it has the engine, the ID engine in the app. I am testing that constantly, but maybe I don't even have the authentication to verify moths. I don't know, or it doesn't appear because it was a Spring module, and that's an interesting thing.
Yeah, so we can add
a lot of things I cannot do with moths because that was a Spring project addition, and
yeah. Well, if there's any, well, we'd very much welcome you reviewing the moth data. So we can set you up with that role in the system, because maybe at the moment you've just got access to the butterfly data.
Not just me, but also there is the curator for the museum, which would be very important for the moth collection.
Yeah.
So
great if you could send us
I write
contact.
you an email then with the names, and then we can get that. Okay, good. Number two, I checked the previously I checked the rules, and there were many rules which were obviously out of time period, for example, the flight period, which were wrong for Hungary. Can we modify the rule? Because at that time you said no. So can we?
Yeah, I think we
It's the rule.
Yeah, we don't want to do it too often, but I think we can. I think it's probably timely to refresh those rules. The other thing I forgot to mention actually is there's a bit of intelligence in the system that if you accept records, say where the rules have been failed, the system will update itself or sort of learn that if I can't remember the exact numbers, but if you confirm the occurrence from a location of maybe half a dozen records, the system will then update the rules in the background. But we might want to formally review these rules, particularly in parts of Europe where the knowledge is changing quite rapidly. That I think the rules can get updated quite quickly.
Okay, and the third question is, I'm just checking behind the Zoom window. I just went into the verification pane, and I'm just watching our data now. This is the Oedipus data, and of course it is flagged, although I didn't do anything, but because of the list which has its problems, it's flagged as confidential because of the species itself, as usual. And there was this issue now with the location. It's not precise location formally. I know that this has been fixed because we have many problems with, especially with Oedipus, when we downloaded the data from the system, and we had all kind of bullshit because of the masking of the actual location. So it was just on a larger grid. Now it has been fixed. At last time it was. I haven't checked it, but this is fixed now, yes? If we download the data.
I understand. Yeah, my understanding is that's been resolved.
I will check it anyway. I will do a check.
And you should, as a verifier, you should see the full records, the full record details. So the blurring and the confidentiality is typically for the general view of the records.
Okay, so that's confidential. Doesn't really mean for us verifiers anything. Yes, because that's this little triangle, red triangle saying it's confidential. Okay, good.
Yeah, I'd need to double check that, but that's my understanding.
Okay.
Yeah, I think the sensitive data will appear anyway on the verification. I think, yeah. Okay, Andreas, thank you so much for your question. I don't know if there are other questions or if we can move forward and I can show you exactly how is the system on the website. If not, just place it on the chat. I think the majority of you are already as a verifier. Okay, if I enter on eBMS, you will see something similar. Is my account. Then I see more things that you can see. So, for example, here on the upper side, but don't worry. It's just I want to see that if you have the role as a verifier, you should have this tab of verification, and then be able to see the verification page and how to do exactly. We can set up any filter. I was checking, Andreas, I think you are there. If I go, here are all our verifiers. So here I have Andreas and then I set up for what exactly, but we could check if it's not working or whatever. But we could check here anything, any person, any user on eBMS that you tell us exactly for what exactly they should do the verification. So we could define for what exactly, so only butterflies or only moths. So that is a possibility. And also to define exactly for where. We could do it just for a country. So for example, Italy, and then all the records will be sorted to this country, but we could do it even to another level, to the provinces and regions per country. For example, in my country, in Spain, is really useful. So then we could apply for a region specifically on a country. So we could define it as you tell us. And also from where it's coming, no? So if it's coming from only moth traps or 15-minute counts, we could also set up the filter in that way. And if then what we define on the filter is what you will see in the verification system. So in this case, I have I can see all the records submitted to eBMS. And as David was explaining to you, just to show you a little bit, maybe you are really familiar, but sorry for duplicating the information. But when you select one of the records, then all these buttons will appear here, is highlighting here in red where is exactly that record, and tell you more and more information on when it was submitted, in which here it was, for example, a transect, and more. And then from all these buttons that David was explaining, here is the status of that record. So at the moment it's pending, and you could define if you accept it or reject it directly. We normally do the double check, accept it as a correct when you have pictures. So in the case of butterflies, we normally don't have so many pictures, but on the case of moths, you will see you have a lot of pictures, and then in that case, you could see it even closer and define that is correct or not. More things here, you have different filters. So which records are on pending, or which one has been accepted. So for example, we will see many from Portugal, no? Here on the map is nice because you could see where it has been accepted records. So in Denmark and Portugal, that we are going to show you now. Also in Lithuania, really nice. And here is the status of that record, no? That has been accepted, and it has the picture. And you can play quite a lot with this table. So you could define, you can search as, do you want in all these searching bars? So you could look for a species perfectly. You will find, we will see tons of those. And you could see here also the rules that David was mentioning, no? That is applied directly. In this case, we have pictures because I set up like that. And here you can see on the map all the Pieris wrapper records that has been submitted. And in this case is pending, no? So you could check the picture. It's not the best, but you could almost say what is the species. And you could always accept it, or even in the moment that you need, okay, this is not Pieris rape, it's Pieris napi, you could redetermine and define that is another species. Here I will blur all this information about people here, but you have the emails of any one of the recorders, so you can submit them emails to tell them this is you have an issue with one of the records. You can even comment it on the record, or see the recorder experience, if that person has been recording for so many years. In this case, not just for 2025, but that is useful information for you. Well, interesting things here. I reset everything. But as all the tables on eBMS, this second icon, the tool icon, is defining the table on what you exactly want to see. So for example, I'm not interested on the last update or on the common species. I unclick it. So you could include in that table wherever you want. So for example, day of the year or any other information. I was showing image classifying agreement. This can be interesting for the moths, for example. So you could see here in the moths if that can be passed an image classifier, no? So in this case, it's telling you yes. So that could help you to define the final status of this record. But you can always come back to the default structure of the table. Another thing that I think is really useful is this top icon, that if you click on it, you are able to to verify records, several at the same time, no? So for example, you know a species and then you tick all at the same time. In this case, for the 30 that appear on this page, but you could go even to more, to 120, and then you tick all and then you accept it. So then it could help you on the process of doing the verification. So I don't know if there are specific questions for this part. So anyone is welcome to open the mic or mention something. If not, we move forward to some examples of how they do it in other countries. So I don't know if you were all familiar. I think the majority of you, yes. But I think it's nice to discover how exactly the system works. I stopped sharing.
The only other thing I think, well, maybe you didn't mention it, there's the option to send the record to somebody else as well, another expert.
To another expert is possible. Ah, yeah.
Cristina.
Yeah, Goran.
Sorry, Cristina, I have a question. Some of the records are crossed out. So what is the meaning of this?
Are crossed out?
Yeah, they're absence records. So it's
okay.
I think they're probably coming from single species 15-minute counts, probably.
Yes.
So you've looked for species that's not there. So it's an absence record.
Okay, thanks.
So there probably aren't many of those, but that's what they are if they're crossed through.
Yeah, thank you very much.
Yeah, you can see it sometimes on the table, or could be also that someone deleted, no? So it could be possible that that record was not correct.
No, I think it deleted, it won't appear or disappear, but I think, yeah, I'm pretty sure they're all absence records. So that,
Okay.
yeah, there won't be many of them, but, and how you verify them is an interesting question, but yeah.
Okay, great. Thanks. Thank you, Goran, for the questions. Then we move forward to Andreas. Let me know.
All right. Yep. I'll just try to share my screen then.
Very short. Yeah.
No, Andreas. Sorry,
sorry.
I was Andreas
Yeah.
that has a question. Sorry.
Similar names. Sorry.
Andreas. Yeah.
Okay. Sorry. Andreas is talking now.
No, you can go. Yeah.
Oh, okay. Just one thing. I know that it doesn't appear here, the training records, which we had problems with when someone clicked it on, and also that happened that didn't even do that, but there was some bug. So is there an option for verifiers or local administrators to change the record, or it will remain in the same way that we have to go to you, David, or writing to you when we want to change training records into actual records?
Yeah, currently there's no way of doing it on the online system.
Okay.
But that is a good suggestion to flip them to being non-training.
That happens sometimes. Yeah. Okay.
Yeah.
Yeah, we know.
Yeah. We'll think about that. We'll add it to the list. But if you've got any that you know, yeah, email us with the details and we can switch them to live records.
Yeah, thank you, Andreas. Indeed, it could be interesting that the coordinators or the verifiers could do that. So we will investigate. Andrea, sorry I say it, but I put it on the agenda that Elder will go first, if you don't mind. Okay, so Elder, if you can go.
Yes. Hello, everyone. Thank you for the opportunity. I'm going to just introduce really briefly what we've been doing down here in the corner with moths, with the Portuguese moth recording scheme. So I prepared a short presentation. Just I'm going to share it. Just a sec. Okay. Okay, is it all right?
No, now we can see it. Yeah. Thanks.
Okay. So just to introduce you before I move on to my colleague João, who's going to talk about the verification process, just to let everybody know what's the Portuguese moth recording scheme like and what we've been doing over the last few years. So it began in 2021 as an informal group, a group of 6 moth enthusiasts that we trap in a regular basis. So we want to do something coordinated. And then we started thinking about a national scheme, a volunteer base. And then in 2023, we created as an organization, as the project grew, a little bit as a surprise. We weren't sure if the project could grow with moths in Portugal. Was not a very known thing. So, but fortunately things went well. And then later on, we established the collaboration protocol between the Butterfly Conservation Europe and the project itself. The objectives was mainly to establish a Portuguese network of moth monitoring using citizen science, and also to support the creation of knowledge and gathering of data. There was some moth samplings over the country over the years done by many people, even from visitors from the outside that came to Portugal on their holidays to trap. But was always very not on a reliable basis. So this we wanted to produce data on a monthly basis, almost on a weekly basis. So that's why we decided to go ahead with this project. And also another big aspect, which is a key for the project to grow as well, was also to raise awareness of this group for moths with the public itself. Since was barely in existence in Portugal as a public subject, let's say. People just know moths for their wardrobes and that's it, or as pests in crops. So we wanted to change the perspective on that a little bit. The methodology was it's basically open to anyone regardless of their experience, basically because of the tools we set in place to help people starting. And also the verification process is a key factor into that. We use non-lethal light traps, especially with fixed locations that we call stations or estações in Portuguese. The samplings can be done in any kind of habitat. We have, for example, moth stations in balconies, in gardens, more remote areas, protected areas. And we allow 2 types of trapping methods. One is unattended and attended, which we've been using moth traps, like a bucket with a funnel. And also we allowed some stations to work with a sheet as well. The sampling is minimum sampling to participate in the project, at least sampling once a month. Most stations, they do more sessions than this. Some stations, they do weekly traps, sometimes every 2 or 3 days. For the attended traps, the minimum effort is 3 hours around the sheet. And for the light traps is usually all night long. We start using later on in the project. In the first years, we used our own database, which did the job, but since was not online, was a little bit cumbersome. So with the rise of the integration into the Butterfly account app and the eBMS, the thing was much more useful and reliable to collect data and also for us to review the data. So all individuals are identified to the species level whenever possible, of course, and the number of individuals counted too. We produced over the years several tools to support the project. One of them is the methods manual that we send to everybody who want to start collecting information. We also build several video tutorials that we keep on our webpage as well, how to sample, how to use the app, how to build your own moth trap as well. So we have plenty of videos available. We also did last year, we published the first comprehensive photographic guide of the macro moths of continental Portugal, which revealed to be a very useful tool for people that just starting, or even for educational purposes with when you have sessions with public as well. So this has been a very interesting step forward in the project as well. So we build up this book with photographs for the majority of the almost 1,000 species that we have. Also with the distribution maps for Portugal, as we know it so far, and also flight periods too. So, and also have the difficulty of identification. So people can be aware how challenging it can be to identify that moth. We also have a quarterly, it used to be monthly newsletter. Now we have to move it into a quarterly because it's a lot of work. It's a very complex, almost a magazine style, that it's key feature to spread the word on what we're doing. Because one of the things we really wanted to do is if we started this volunteer-based project, we want to start giving feedback almost immediately and constantly for the people who are working in the project with some results at least. We also create a private group on Facebook where everybody that has a moth station can participate and exchange information. We do regular online and in-person meetings and talks as well. And also a step forward was the development of a lead trap, an affordable lead trap for the Portuguese reality too. One of the things we figured out right away was something that was kind of in the middle of someone that want to start the project was to build the trap itself. It would be a turn off for a lot of people, especially when they had to deal with some electrics, even if not complicated, but for the majority of people, they would just say no or abandon the project because of that. So we had to create a solution to grab and reach those people, not to give up on the project right away. And as the results, we have currently about 80 stations all over the country. And here you have the stations. This is actually an interactive map on our website. So if you click in each one of these dots, you'll see more information about the station. And as you can see, this follows the demography of Portugal as well. So all the population is concentrated on the coastline and in the south coast, and the distribution of moth traps follows that pattern. We've been trying to fight this and trying to promote the project in more inland areas where there's a lot of biodiversity as well and very interesting habitats. So far, we have a little bit above 65,000 validated records over the 2 years that we've been using the eBMS. More than 1,000 species of moths. We found several new species for Portugal in the project itself, and we updated a lot of the distribution map range for a lot of the macro moths especially. So this is just the looks of our quarterly newsletter, some of the latest covers. And they're all available to download on our website. Also, you can subscribe freely and you get it on your email. And we cover from identification articles. We have several people writing for the newsletter. And also even we go into depth into more like genitalia dissection themes, but even more broad themes about moths. And we actually also launched this year in April, so it's quite recently, a scientific magazine. This is run by Martin Corley. It's called Artimelia, and was built basically to fill a little bit of a niche of publishing scientific articles on Iberian Lepidoptera, focus on moths mostly. So we're giving the first steps on this. It's a biannual, gonna 2 issues a year. So we just started, but it's been very interesting journey. And that's it. I invite you all to visit our website as well. We have plenty of information there. And I'm available if you have any questions. I don't want to take too long with my presentation too. Thank you, everyone.
Thank you so much, Adler. It was really interesting. I think there are two. Well, there is a question about the methodology manual, and João shared the GBIF project, of the Portuguese moth monitoring.
Oh yeah. We also published every year we publish the set of data from the previous year, so we have 1 year of delay on the GBIF as well. So we release all the information right away to be stored because it needs to be verified and validated carefully before it's set on GBIF as well.
Sure. Lars is really asking for the specific manual where you explain how to really collect, no? How to set up the trap. And so if you
Yeah, it's kind of the basis of how to put up the traps, how the project works. So I can send to Lars by email later on if he wants it.
Or you
on
can
a PDF.
leave it on the
Oh yeah, or
chat.
over there.
It would be
Yeah,
great
yeah.
for the rest. Thank
Sure,
you.
sure.
Really interesting. David Roy wants to talk. Yeah.
Yeah, thanks. That's great. It's amazing what you've done in a short space of time. I was wondering, maybe it's in the manual, but I was interested to see that you've given some advice on how to build your own traps, because I think that can be a barrier to people getting involved. And I think if we've got an easier option for do-it-yourself traps that could be built, then I think that would open up more interest in other
Yeah, in
places
the
of Europe as
manual
well.
we have some modules that people can build, and also different types of bulbs that you can use. That it's always very complicated. Someone starting which type of bulb will I choose, and then the energy options to support that bulb. So have it in my garden or going far away from my house. What can I do? So it's always a tricky and complicated thing. That's why we end up developing a little bit following the model that you had on the Netherlands with a bucket with a power bank.
Yeah.
So that's really useful.
Great, thanks.
Great. Thank you so much. So I think we move forward to fit on the time. Thank you, Alder. It was really great to listen to the project. João, are you ready to continue with the Portuguese team?
Yes, I hope so. Let me share my screen. Hello, everyone. Sorry. Are you seeing?
Yeah, we were seeing your.
Okay, was the right screen?
It was
Yes.
on eBMS website.
Okay,
Yeah.
perfect. Thank you.
Maybe if you can zoom in a little bit, it would be great.
Okay, it's better?
Yeah, great.
Well, okay,
Thanks.
thank you. So moving forward to verification segment. So that most of the tools were already presented by David and Cristina. So I will just do like an exposition of how we do it. So right now we have like more than 200 records pending of validation. If we search here for the accepted records, we can see that we already have more than 65,000 records validated in the last 3 years. It's quite a work and at a daily basis. So, but starting with a sampling session, I will start, for example, with a random recorder that I search here. So, okay, well, how I start the process? I usually pick up one sampling event from a particular recorder. Here I started with the recorder Joanna, and I know that here we have 25 records, and I usually start by enabling multiple records selecting. I tick all the records, and then I just check which are correct or not. For example, this one is correct, but I will search for one that is not. For this one is identified as unknown, so I should untick this one, and I will not go for most of them. So I will just select these two that are correct identified, and then when we have pictures, we select this one, the double tick mark. And when we don't have pictures, but we know the experience of the recorder, or we have more information, but by other platforms, for example, by calling the person in the responsible for these records, or by an email or other mean, we usually only select the one tick mark just to differentiate the record that we have photographic evidence from the records that we don't have that evidence. We virtually only use these two categories for our records, because when the record is not possible to validate, we only reduce the identification level for the level that we can ensure from the photo. For example, this one is a tortoised from the genus Lobesia. We can't move much forward than that to the species level, so we just identify this record by the genus level, and then after the platform updates, we select the double tick mark because we have photographic evidence, even that the identification is only at the genus level. A tricky part from this platform is that we also have here Lobesia species from the same sampling event on the same locality from the same recorder, and the platform does not sum up these records automatically. So how we deal with that? We open we clicked here and open the page from this specific sampling event. We click here to show the full list of records, and then we show, for example, here we can is another example. We have two different species associated with the same record. So this one is correct, it's Pseudops prasinana. We remove this one and reduce one in the quantity of the record. Is a very difficult to keep this work done, but going to Lobesia, we just need to search for in this case we have two records of the same genus that is not possible to identify to the species level, so we just need to concatenate the records by removing one of them and add the number of the previous record to the next one. In this sampling event with only 25 records, this is quite easy to deal with. But in sampling events with hundreds of records that we usually have mainly in south of Portugal in the middle of the summer or in the late spring, it's quite difficult to keep the work done for mainly in the weekends where we have like 20 recorders sampling at the same time. So if this problem could be solved, it would be very welcome to increase the efficiency of the validation process. Assuming that everything is okay, then we just need to save, and in a few seconds or a few minutes time, the record will be updated in the validation menu. You can see that the quality of the photos is not perfect in most cases, but for example, I will search here for another recorder that is quite dedicated, and the quality of the pictures is much better. This is a very, very small moth, and the quality of the picture is quite well, it's correctly identified. And this recorder in particular, but others too, do dissection. So, for example, in some records like this one of Indotinia marginana, we have not only the picture of the moth, but also the dissection. Here is a male dissection, the main apparatus and the Ediagos. And when we have this kind of information, we ask the recorders to, in the occurrence comment, let a note identify that this specimen or this record was supported by genitalia dissection, and hopefully in most cases the recorders put the sex of the individual that supported this identification. This is very important because when we are converting the data for publishing it on SBIF, one of the columns is identification remarks, and there we put the information to that this record was supported by dissection, and this is very important for many species or very many cryptic species that need this kind of identification method to be reliable. And when we don't have it, for example, if this species was not this would not be dissected, the identification could not be accepted, and I will reduce the identification to the genus level, to Indotinia in this case, and validate it in any way. So this one is also okay. Let me see if I have another tricky case here. So you can see that the quality of the pictures is much better than the previous recorder. It's the dealing with monitoring schemes is this is one aspect of that, dealing with very different approaches from the volunteers in the recording data. And here is another case of dissection. I will not play it because I need to see if this section is well performed and the identification is okay. Here I will also remove. I need to check that later carefully. This one is okay. I will validate. And I will just try another recorder. For example, these records were those that Cristina was showing in her presentation. And for example, we have here two records of the same species under different species. Here, Eupithecea pulcellata, and here, it's the same species but under a different name. And if those merged automatically when I change this identification, that would be perfect, but it's not the case yet. And I think that's it. Well, at least the main of the work. When we can't identify at all the species that we are seeing, for example, with a very poor picture or a very damaged specimen, we usually remove the record. For example, I think I don't have any example of that here, but usually very small moths like nepticolids or gracilarids that are very damaged within the trap, or that the photo is very pure. We usually remove that record because we can't, in some cases, we can't say that it is really a moth or a trichoptera or another insect, for example. So we just remove that noise from the platform, that it's not really important. Another aspect before asking for questions is that our project is focused on macromoth, but many recorders also report micromoth, mainly the macro micromoth. So like this one, like Phomia social is a micromoth, is a pyralid, but is more or less bigger than many geometrids and some noctuids, small noctuids, of course. And many records report them, and we validate, if possible, we validate those records because we should not discard such records that are perfectly fine to be included in the platform. And I think I went through the most of the important parts. I don't know if someone has some questions regarding this. Feel free.
Thank you so much, João. It was really nice to see how exactly you do. Is impressive also to see that you have even dissections of the genitalia attached. I never saw that on the verification, so really nice to showing us to everyone how do you do it. It's awesome, the work of the Portuguese team. Thank you so much, guys. It was just one mention, Lars, about the common names for the moths are not correct. Sometimes it appears Swedish or German names to the moths. We are working on that, on applying the common names also, because you are including not Portuguese names. So we should work on that, improving the names for the moths. And I think it was another question. No, you mentioned the possibility of collapsing, no, the species that there are with the same name or genus. Something I'm sure David is taking note of that, and we will try to include it.
Yeah, I hadn't realized. Yeah, I can see the problem now. So I'll talk to the developers and see whether we can have some automatic way of doing that.
Perfect.
Yeah, it's the good part of using it, and we see what we really need. Elder, do you want to mention?
just to add one thing also to what David said before, that to have rules for the moths will be automatic rules will be great as well. If we could work on that in the future, we have already, for most of our macro moths, we have, for the majority, I would say, or all of them actually, we have already some interesting information on distribution and also flight periods that we could easily build some filters also to apply, at least for the Portuguese reality. And that could help a lot the reviewers to flag species that would fall outside those rules. Would be nice to work on that in the future if possible.
I think we take into account also because we need to include it for the whole of Europe. So yeah, we will see. But yeah, thank you, Helder. Andreas, a short question.
Yes, actually, I haven't checked the data structure of the downloads. What happens with the unapproved records?
Good question.
We can filter that. For example, we
Okay,
only
okay. But when you download, for example, all of your records for your BMS, all of the records are there. Is there David, is there a flag, or is there a field there saying that verified?
yes.
There, yes.
It's called record status, I think. So
Yeah,
it should
there
be
is.
part
Okay.
of the download.
Thank you.
Yeah.
I couldn't.
So I
can't
think you get everything, including the rejected records.
I think there is a common column specifying the status of that record, I think.
Yeah.
Yeah. Good. Any other question regarding the moth identification? I think it was excellent and we have it recorded, so it's so great to have it and then use it later. Lars?
Hi. Yeah, I'm a little bit curious about the ownership of the records. If they are validated and changed to something, can a sort of stubborn volunteer go and change it back? I know, for instance, in the database here in Sweden, we or the species portal here, they assign the ownership of the data to the database, and once the volunteer has done that, then it disappears from what they can change. But in principle, you could have people that go in and change things back, even that they insist that they know what it is, even though it isn't. So I'm just curious what happens with the record after it is submitted, if the volunteer can change it or not.
In theory, it can change, and you could get this flip-flopping, but in reality, we've never seen it, I don't think, in the UK situation. So yeah, the records remain open to editing. I don't know if the Portuguese have seen it.
When a recorder changes the and the record already validated, the record comes again to the validation phase. Even, for example, the section specimens the example that I shared, if a recorder later on dissect that specimen and change the comment saying that that specimen was dissected, so only changing the comment associated with the record, that record also comes again to the validation phase. So any alteration to the records will again pass through the validators.
Yeah, I was just about to ask that if the database resets the validation status, if something happens to a record, then everything is fine.
Great, yeah. So nice to know that that is updated. So any change to the record will appear anyway. Thank you so much. Let's move to Andreas from Denmark to present the examples on how to do the validations of butterflies.
All right,
Yeah.
yes.
Yeah.
Just share my screen.
He is part of the Denmark BMS. So yeah, thank you for presenting today. We can see it not in presentation mode yet.
And right there.
Yeah, now it's
Right,
correct.
does it look good?
Yeah.
Great, thanks. Yeah, so I'm here presenting the Danish BMS and the verification process. I think Anna is still here also, so I guess she'll also be available for questions, or yeah. Moving on into the presentation. So we're relatively new butterfly monitoring scheme. Just to give you an overview, we started in June 2023, so very young, but we already have 98 unique transects, so that's really great. And you can see from the figure the coverage across the country. Unfortunately, of course, our transects are correlated with population density, so we have a problem reaching some of the outer regions, and often the butterfly diversity is not correlated with population density. So there are a couple of species that we're not catching in our transects yet. But it's still growing. So I just checked May as we finished last month, and in 2024 we had 160 transect walks, and this year we had 259. So we're still growing. Really happy about that. And you can see from the colors from the figure also that most of the transects continued from 2023 into 2024. A couple were abandoned, but we've actually been able to repopulate some of them also with new volunteers. So that's really great. Yeah. Going into the verification, we started verifying records last autumn when we were made aware of the system. And we try to verify all records, so not just removing the mistakes, but also confirming the records that are correct. And that helps for some of the things that we want to do with that data then when we want to filter it later. We also really try to follow up with volunteers. It's mostly done through email. And it's my impression that they're really happy to get feedback on their records so that they can also improve their skills. Often it's cases where people are not aware of when a flight period starts or something like that, and then actually that ends up possibly making my work easier for the next year when people improve their identification skills. Yeah. Yes. So as I said, we started last autumn, and so we actually did a yearly verification for all 2024 data and the 2023 data that was also in there. But we've been sort of switching to a monthly verification because so the figures that you see on the right are the ones that we share with the volunteers on a monthly basis in our Facebook group to give them an update on how they are doing in the project and try to motivate them. And obviously we don't want any misidentifications in these graphs. We don't want any species to occur in those that have obviously not been seen during that month or something. So I try to pick out the wrong identifications on a monthly basis, and then we might do the confirmations of correct records at the end of the year. Yeah. Yeah, and so just also included here, that was actually a question about this before. So this is the table that we pull out from the BMS website, and you can actually see those columns that we just talked about with the verification status there, and that really helps with filtering the records then for these figures and the other stuff that we want to use the data for. I think, however, it's only included at this point in the transect data. I might be wrong because I haven't looked at the 15-minute data for a while, but I think it might still be missing in that one. Yes. So our experience with these automatic filters is that most records fail the filters on phenology. I think the icon is like, you see my cursor, right? Yeah. So I think the icon is different. It's a ruler here. I think it was a sort of a calendar icon when David showed it, but the ruler here means that it has failed on phenology, and they seem relatively strict right now. But I I guess I like that because then I know that I have been through all possible mistakes. It's better it's that way around than too few getting caught by the filters at this point. Yeah, so I see very few or records that fail on the geography, and I think that is because it's quite coarse, and as David mentioned, we have these coarse polygons around the coastlines. So some of our transects actually fall on the outside of the Denmark polygon, and then all of them ends up failing on geography from that specific transect. Yeah, so it's not perfect right now. I don't think there's actually any species where it catches species out of the distribution so far. So what we do to supplement that right now is that we have good databases in Denmark so even though the BMS is young, we have a lot of records from other databases that don't record effort when they observe. So this is the distribution map of Cyanaris semiargus, and we take those records then, and I put them into an R script that will then generate a distribution and then check all the records against that distribution. So right here you can see that in the transect so far we have 2 records of this butterfly species, and they both fall inside of that distribution. So but yeah, that gives me a list with records that I can then double check and then we do that for the red listed species and for species that otherwise have a restricted range within Denmark. For example, we have common species like Tumilacus sylvestris, which is really common, but it's basically missing from the entire sealand. So we check those also and get a list of that, so we can quickly have an overview of what we need to process. Yeah. Yes, and so as David mentioned, we also publish the data to GBEF. And for this, the verification is also very useful because it gets filtered for only the accepted records. They end up in GBEF, and then we also wanted to publish our data in the national database. So that's what you see over here on the right. And they fetch the data from GBEF. So that means that if we just push verified data to GBEF, then also only verified data will end up in the national database. So it's very practical in that way. All right, so that's what I had to tell you about the Danish verification.
Thank you so much, Andreas. It was really nice to listen to how you do it, even it was really young when you started. Yeah, doing a marvelous job. Thank you so much. We still see your screen. It's black. Yeah, thanks. Really nice. So I don't know if there is any question specifically about the butterflies. Yeah, it's a different case, no? Because you don't have so many pictures, I imagine. So you are working more with the distribution of the species and the time period.
Exactly, and then we're lucky we also know a lot of our observers because the community obviously didn't start first in 2023. So we know a lot of these people, we know their experience, and we also put emphasis on that when we evaluate the records.
Yeah. So it makes sense. It's a different approach compared to the moths, but yeah, it's also working and it's nice. So yes, I don't see any questions on the chat. It was saying, yeah, really nice increment of the transects in Denmark. Irma mentioned it. It's really great to see how the Denmark BMS is growing. Yeah, if there are not any other questions, I think it was really great to see the differences in some countries, how you deal with that, how to use the verification system. So I hope is giving some tips how to use it. Anyway, this video is recorded, so I will post it and share it with all of you and with people that has not been present here, so then we can refresh it anytime you want. And I just to mention that is everything works thanks to teams. For example, in Portugal, is not only João and Elder. I know there are, I don't know how many verifiers, at least 4 or 6 people. So it's not only depending on one person. So
Yeah,
we
there's
combine
four now. Yeah,
four
four. Yeah.
4 people already in verifying, and you can see that they have only 200 pending now, but they have been doing a really great job before. So now at the moment for a country starting from the scratch can be kind of tedious, but yeah, step by step, I think we can get it there. And yeah, let us know to David.me. We can include anyone that you consider to be verifier for your country or region, so we can do it and help on that process of verification. And any questions or suggestions, we have been writing down and trying to improve in the system using it. So yeah, I think it's all for me. We finished a few minutes earlier that we mentioned. So yeah, thank you so much for being here, the hour and a half almost. And we keep in touch and have a lovely butterfly and moth season. We are really on the peak, so enjoy it so much.
Thanks,
So-
Cristina. Thanks for
Thank you.
organising.
Thank you.
Yeah.
Bye.
Thank you
Nice
so
to see
much.
everybody.
Thank you.
Thank you.
Take
Enjoy the
care.
summer with all the butterflies.
Hmm.
Bye bye.